Okay, so we are on to chapter 12, you guys. We are getting closer and we are on the downswing of this course. So, again, this is the application stuff and it's so much fun. So, this is going to be about chapter 12, which is on drugs, microbes, and host, which would be the elements of chemotherapy. So, the principles of antimicrobial therapy are that we need to administer a drug to an infected person, destroys the ineffective agent without harming the host cell. Sorry, I need to grab my book over here. All right, and then the antimicrobial drugs then are produced naturally, or they can also be produced synthetically. And we talked a little bit about taking over a host so that we can make these other organisms make the things that we need to be made. Okay, so that would be what we're talking about here. So for the origins of antimicrobial drugs, what we have is antibiotics. And antibiotics are common metabolic products of aerobic bacteria and fungi. And so these bacteria would be in the genre of Streptomyces and Bacillus, and the molds would be in the genre of Penicillium and Cephalosporium. And by inhibiting the other microbes that are in that same habitat, what can happen is that the antibiotic producers are going to have less competition for nutrients and for space, right? So they don't have to compete to grow. We just let them grow, okay? So then we can get more bang for our buck. So this is a fun little table of selected microbial sources for antibiotics. And so this is table 12.2 in the current version of the book, but you can see that penicillin is made from mold, cephalosporin is also a mold, monospora is bacteria, bacillus is bacteria, and what these can make would be penicillin, cephalosporins, gentamicin, bacitracin, right? A lot of things that we use daily, right, in medicine. So, talking about interactions that can occur between drugs and the microbes, antimicrobial drugs should be selectively toxic. And that means that they can kill or inhibit just those selected microbial cells without simultaneously damaging the host tissues. So, like if I get an infection, right, and I need to take an antibiotic, I don't want it to kill all of my good cells, right? I just want it to target the bad guys. So as the characteristics of the infectious agent become more similar to the vertebrate host cell, this complete selective toxicity becomes more difficult to achieve, and therefore more side effects can be seen. So when we're talking about the mechanisms of drug action, this is figure 12.2. And so this is showing you the major targets of the drugs that are acting on those bacterial cells. And each mode of action is going to correlate with the numbered section where it's discussed in the text. So we'll continue on with that lecture later. But the cell wall inhibitors are going to work to block the making and repair of and these would be like your penicillins, your vancomycins, your Bacitracin, right? Your all these guys, cycloserine, etc. The ones that target cell membranes and can cause the loss of selective permeability would be your polymyxins. The ones that are going to be targeting DNA or RNA and therefore inhibiting replication, transcription, etc., these would be like the ciprofloxacins or the rifampins, right? And then we have protein synthesis inhibitors that can act on the ribosomes, and we can even target which part of the ribosome that we interact with and inhibit. And finally, we have metabolic pathways and products that can block the pathways and inhibit the metabolism, which means that that cell is going to die. And these would be like your sulfa drugs and your trimethoprim. So this is a fun little summary table of your drug mechanisms, okay? And these are the mechanisms of drug effects on major categories. And so this, again, is going to be in your book, or else you can just blow this up. And so in the current version of the book, this is broken into more of the subcategory, but these slides were sent to you, so you can go and you can blow this up, okay? So talking about the spectrum, what is the spectrum of an antimicrobic drug? Well, the spectrum would be the range of activity of that drug. So if it's a narrow spectrum, then that means that it's going to be effective on a small range of microbes, and it's going to be targeting specific cell components that are found only in certain microbes, okay? And then we have broad spectrum, and these are the ones that have the greatest range of effect, right, and activity. And so these target cell components that are common to most pathogens, such as ribosomes. Most bacterial cell walls contain peptoglycan. So this is kind of like an easy target, okay? Penicillins and cephalosporins, these are going to block the synthesis of the peptoglycan. Therefore, it causes the cell wall to lyse. And lyse is just a fancy word meaning to break down, okay? So these are active on young and growing cells, and therefore we can really stop the growth of these microbes. Penicillins that do not penetrate the outer membrane and therefore are less effective against gram-negative bacteria are present as well. Broad-spectrum penicillins and cephalosporins can cross the cell walls of gram-negative bacteria. All right, so when we're looking at the specific activity of penicillin, this you can be seeing in figure 12.3, okay? So up here in A, this would be, so the whole overall arching of the name of this diagram is effects of penicillin on gram-positive cell walls. And so in A, up here, what's happening is this is a normal untreated cell. The left and center views are staphylococcus cells, and the right view is the molecular structure of peptoglycan with chains of NAM, which is the N-muromeric acid, and NAG, which is N-glucosamide. And these are linked together by cross bridges, okay? And you can see these here, and those little green guys are your cross linkages. And then what we can do is we can have an exposure to penicillin. Then what happens is these weak points that lack that peptoglycan are going to kind of break, and then a fragment of peptoglycan can break off. The membrane here can bulge out as water diffuses into the cell. Since water is going to be going in, it kind of makes the cell lyse or explode, right? So then you can still see here that the cell is going to break and lyse, okay? And so the effects of treatment with penicillin are shown here on the left side. Oh, I think I already, yep, we already covered that. Okay. And then, yeah, so basically this is like a figure drawing, and then you can see the cells actually exploding there, okay? So antimicrobial drugs that are going to disrupt the cell membrane function are going to work by having a cell with a damaged membrane. This is going to cause it to die from the disruption or the breaking, the interruption of metabolism or lysis or that breakdown. And these drugs have specificity for a particular microbial group. These are based on differences in types of lipids in their cell membranes. Polymyxins, these are going to interact with the phospholipids and cause leakage. So you can leak in and out, right? And then this would be particularly effective in the gram-negative bacteria, where amphotericin B and nystatin, these both are going to form complexes with sterols on the fungal membranes, which therefore can cause leakage. So this is a close-up of the activity of polymyxins. Okay, and so this is figure 12.4 in your book. And so the polymyxin is going to bind to the outer and the cell membranes of gram-negative bacteria, and this is going to end up creating abnormal openings that you can see here that cause the leakage or the intake of fluid, right? And therefore, because of that, the cell is going to kind of explode or lyse. So that's how those work. Moving on to drugs that affect the nucleic acid synthesis, and remember this is DNA. DNA is deoxyribonucleic acid, right? So the actual genes of the cell. So what happens is these drugs may work on blocking the synthesis of nucleotides. It can inhibit replication, or it can actually also stop transcription. Chloroquine, which it's really interesting because right now we're in the middle of COVID, and I believe hydroxychloroquine was one of the drugs, and I believe it's in the same family member, right? Anyway, chloroquine is going to bind and cross-link the double helix, And then quinolones, they are going to inhibit those DNA helicases. And remember, the helicases are the ones that unzip the genes, and it makes that double strand into 2 single strands so that those promoters can get in there and start making copies. Also, antiviral drugs that are analogs of purines and pyrimidines. Remember, those are A's and T's and C's and G's. And analogs mean that it's like the same shape, right? So what can happen is they can go in there and they can insert the viral nucleic acid, therefore preventing replication. So moving on to other types of ways that we can affect these, we have drugs that can block protein synthesis. Well, ribosomes of eukaryotes differ in size and structure from prokaryotes. And those antimicrobics usually have a selective action against prokaryotic cells, and they also can damage the eukaryotic mitochondria. Okay, you have the aminoglycosides, which would be like your streptomycin and your gentamicin, and these are going to insert on the sites of the 30S subunits of the ribosome, and this causes the misreading of messenger RNA, therefore a misformed protein, right? We also have tetracyclines, and tetracyclines can block the attachment of that tRNA on the A receptor site and therefore stop further synthesis or making. So this is figure 12.5 in your book, and these are the targets of the 70S ribosome. So these would be the sites of inhibition on the prokaryotic ribosome and the major antibiotics that act on these sites. All of these have the general effect of blocking the protein synthesis, right? And the blockage actions are indicated by X's, okay? So up here, the little triangle, this would be the aminoglycosides. And so this big subunit is the 50S, and the little guy is the 30S subunit. And so what happens is the mRNA is misread, and then the protein is made incorrectly. And so this little mRNA strand is made wrong, okay? And then another one, this here is the chloromenthicol, and this is where what happens is the formation of the peptide bonds is blocked. And so we're going to not be able to make our peptide bonds. And remember, those peptide bonds are what links the peptides together in the primary chain, the primary string of protein, polypeptides that make the polypeptide chain, which therefore folds into an active protein. All right, with these guys, these are your oxalidones. And what happens is this is going to prevent initiation and therefore block ribosome assembly. So you can see nothing is coming out because we're not going to be able to get in and actually make a copy. Tetracyclines down here, this is where that little triangle Star Trek looking thing goes. And this is where tRNA is blocked and no protein is going to be made. Okay, and then the last grouping is erythromycin, and that's this little purple ball there. And this is basically the ribosome is prevented from translocating. And so that would be where we're targeting on the 70S ribosome. This is figure 12.6, and this would be competitive inhibition as a mode of action. And so basically, again, drugs can affect this metabolic pathway. And so sulfonamides and trimethylprim is going to block the enzymes that are required for the tetrahydrofolate synthesis that is needed for the RNA synthesis. And so what happens here is that up here in A, the metabolic pathway needed to synthesize or make the tetrahydrofolate acid, or THFA, okay, is going to contain 2 enzymes that are chemotherapeutic agents. And under normal conditions, PBA or PABA acts as a substitute for the enzyme pyridine synthase, and this is the product of which is needed for the eventual production of folic acid. Okay, and so only certain bacteria can perform this step, and basically this is that tetrafolic acid, and otherwise known as folic acid, and this branches off to make your folic acid derivatives. Okay, so what happens down here is that the formula of the sulfonamide molecule is compared alongside of PBA. So that part is missing. It's actually right here. Okay, so you can see the very similar shape. And so this is why it's competitive inhibition. So we'll go back to that in a second. But basically, so despite the similarity in overall shape, the sulfa drugs cannot be used to make folic acid, okay? And so over here, the sulfa drug molecules can insert into the active site in the enzyme that would ordinarily fit the PABA. And then this will work only when the sulfa molecules are more prevalent. Okay, and they can outcompete the molecules, okay? So when we're looking at different pathways, we have a couple ways to inhibit or stop the interaction of enzymes, right? And the first one is competitive inhibition. The other one is non-competitive, okay? And this one, but they're talking about synergistic effect. So competitive inhibition is exactly that. You are competing, my hair is crazy. You are competing with that active site and you're trying to kind of block the right thing from going in. So I like to say the analogy of you're putting the wrong key into a door lock and it will fit, but it won't open it. So it's stopping the right key from going in there. And so this is what competitive inhibition is, okay? Where the drug competes with the normal substrate for the enzyme's active site. We can also have the synergistic effect. And this is when the effects of a combination of antibiotics are greater than the sum of the effects of the individual antibiotics. So they can work together to be even stronger, okay? Oh, that was moving me. All right. So when we look at the major antimicrobial drug groups, we have antibacterial drugs such as antibiotics, and which can be synthetic drugs. We have antifungal drugs, we have antiprotozoan drugs, and we have antiviral drugs. So they're kind of named for what they do. There are about 260 different antimicrobial drugs that are classified into 20 different drug families. So the ones that can act on the cell wall, and we had that summary table earlier, these would be like your Beta-lactam, and these are your antimicrobials. They all contain a highly reactive 3-carbon, 1-nitrogen ring. The primary mode of action is to interfere with the cell wall synthesis, and this is greater than half of all microbial drugs fall in this class. And penicillins and cephalosporins are the most prominent beta-lactams. So when you talk about penicillin versus the other closely related relatives, okay? So this would be a large group, a large and very diverse group of, I'm trying to find this picture exactly in your book, okay, of compounds that can be synthesized in a lab, right? So we can make a lot of them rather quickly. And these are more economical to obtain since we can grow them on command than through, like, when we use microbial fermentation, we can modify it to a semi-synthetic form. So we kind of make it and it's natural, and then we can modify it and then it makes it synthetic, so then it's semi-synthetic, right? And these all consist of 3 different parts, and that would be your thiazolide ring, right? Your beta-lactam ring, and then also your variable side chain activity. And so the closest figure I can see to this is figure 12.7, and that would be the chemical structure and function of semi-synthetic penicillins. And so all penicillins start with a central molecule, okay, which is going to be this over here, which is the same, and that's called a nucleus. That is going to form the basic framework for that penicillin molecule. And then the nucleus is an inactive penicillin derivative, which is aminopenicillinic acid, and this has a bonding site on the nitrogen on the beta-lactam ring for the addition of a side chain or that R group. And this fine-tunes the end product to create a finished product molecule with a specialized desired action. So here you can see penicillins that have resistance to penicillinase. Okay, so this might be where the figures are a little bit different, but you can see that there are different classes. So the backbone, the guts are the same, but the outward characteristic or that R group is different, kind of like when we were identifying amino acids. So when we're talking about the subgroups and the uses of these penicillins, well, penicillins G and V are the most important natural forms. Penicillin is the drug of choice for gram-positive cocci, like streptococci, and some gram-negative bacteria, such as meningococci and Syphilis spirochete. We also have the semi-synthetic penicillins, which are half natural and half man-made, and these would be like your ampicillin, your carbapenicillin, your amoxicillin, and these are broader spectra, okay, and these would be targeting your gram-negative infections. We also have our penicillinase resistant, and these would be your methicillin, the nafcillin, and the cloxacillin. And then you also can have primary problems with any of these penicillins, which would be allergies, and since penicillin has been around for quite a while, we can have resistant strains of these bacteria as well. So in this table, you can see that there are characteristics of these selected penicillins. So you can go in there and you can you pause this and you can read what's going on, and you can compare and contrast the different types of penicillins that we have. Moving on to the cephalosporins, and these account for about a third of all antibiotics that are administered, okay? And these are synthetically altered beta-lactam structures, okay? And these are relatively broad spectrum. They're resistant to most penicillinases, and they cause fewer allergic reactions. Some are given orally, but many must be administered parenterally. And the generic names have the root words of Ceph, Kef, or Ceph, right? So this is figure 12.8. Okay, so like penicillin, their main nucleus consists of a beta-lactam ring, which is this little dude in red, and a second ring, which is the yellow guy. And however, unlike penicillins, they have two sites for the placement of the R groups, either position 3 or position 7. And this makes it possible for several different generations of molecules with greater versatility in functions and complexity in structures. So cephalosporins specifically, we have 4 generations that exist, and each group are more effective against gram-negatives than the one before. Okay, so we kind of got a little stronger and stronger and stronger. And we also improved the dosing and reduced the amount of side effects. So the first generations are the cephalothin and the cephaloxolin, and this was the most effective against gram-positive cocci and a few gram-negative. Then we went to second generation, and these would be your cephalophore and your cephalinic acid, and these are more effective against gram-negative bacteria. We get even stronger and more detailed. The third generation, that would be cephalexin, and these are your broad spectrum activity that's against the enteric bacteria with beta-lactam. And then your fourth generation would be your cafe prime, and these are the widest range, and these can attack both gram-negative and gram-positive bacteria. Talking about additional beta-lactam drugs, we can have the carbapenems, and the imipenem is a broad spectrum drug for the infections with aerobic and anaerobic pathogens. This is low dose, it's administered orally, and it has few side effects. You also have your monobactams, and this would be like your azarotinums, and these are your narrow spectrum drugs for the infections that are gram-negative or aerobic bacilli. And these can be used for people that are allergic to penicillin, so it gives us options. We also have non-beta-lactam cell wall inhibitors. This would be like your vancomycin, your bacitracin, and your isoniazid. Okay, so Vanco is a narrow spectrum. It's most effective in the treatment of staph infections and in cases of penicillin and methicillin resistance, or if a patient's allergic to penicillin. This is toxic and it is hard to administer, so we use vancomycin very restricted. Okay, we have bacitracin, and this is a narrow spectrum that is produced by a strain of Bacillus subtilis, and this is used in a topical ointment. And then we have the isoniazid, and these work by interfering with the glycolic acid synthesis, and it's used to treat infections such as Mycobacterium TB, tuberculosis. So targeting and narrow that can damage the bacterial cell membranes, these would be like your polymyxins. These are narrow spectrum peptide antibiotics with a unique fatty acid component, and these are used to treat drug-resistant Pseudomonas aeruginosa and severe UTIs. Okay. When we look at drugs that can specifically work on the DNA and RNA, again, this is going off that summary table, these would be like your fluoroquinolones, and these work by binding to the DNA gyrase and the topoisomerase 4. Okay, these are broad spectrum in efficacy, and concerns have arisen regarding the overuse of these quinoline drugs. And the CDC is now recommending careful monitoring of their use to prevent the ciprofloxacin resistance bacteria. Come on, computer, wake up. When we talk about interfering with protein synthesis, these would be the amino, meaning protein, glycosides. And these are composed of one or more amino sugars on amino cyclical 6-carbon ring, and this would bind to the ribosomal subunit. So this is figure 12.9. And it's just saying that the colored portions of the molecule are found in all numbers of this drug class. Okay, so you have that 6-carbon ring that is right here, and then you have the 2-sugar backbone. Okay, the aminoglycosides, these are products of various species of soil actinomycetes, mycetes. It's been a long day. Okay, mycetes. And so actinomycetes in the genre of Streptomyces and Micromonospora. And these are going to be broad spectrum. They inhibit protein synthesis, especially are useful in acting against aerobic gram-negative rods and certain gram-positive bacteria. These guys would be like your streptomycin, which can target the bubonic plague, tularemia, and tuberculosis. Gentamicin, which we use more because it's less toxic and it can be used against gram-negative rods. And then the newer ones would be the tobramycin and the amikacin, and these would help target gram-negative bacteria. We also have our tetracycline antibiotics, and these would be your broad spectrum antibiotics, and these are going to block protein synthesis by binding to those ribosomes. And remember, the ribosomes are on the endoplasmic reticulum, specifically the rough ER, and that is where our proteins are made. So we can use these antibiotics to treat STDs, Rocky Mountain spotted fever, Lyme disease, typhus, acne, and protozoan. So they're very broad, okay? And the generic tetracycline is low in cost, but it is limited by its side effects. And so this is in figure 2.10. And basically, tetracyclines are named for their regular grouping of 4 rings. The several types vary in structure and activity by substitution of the 4 R groups. Okay. I cannot talk today. Chloramphenicol. Okay, and these are potent broad-spectrum drugs with unique nitrobenzene structures. These block peptide bond formations and protein synthesis. These are entirely made through chemical processes, okay? And they are very toxic, which means that we restrict the use, and this can cause irreversible damage to your bone marrow. We use these for the really bad guys, like typhoid fever, brain abscesses, rickets, and chlamydial infections that are quite advanced, okay? So again, this is the structure on figure 12.10, and you can see that it's just a ring structure with the cider. You have microlides and other related antibiotics, and this would be like erythromycin. And this is a large lactone ring that has sugars that attach to the ribosomal 50S units. This is broad spectrum, fairly low toxicity. It's taken orally by your mouth, right? For the mycoplasma pneumonia, Legionnaire's disease, right? Chlamydia, pertussis, diphtheria, and as a prophylactic prior to any intestinal surgeries. And for penicillin resistant, we can use penicillin resistant gonococci, syphilis, and acne. We can use these. And then also we have newer semi-synthetic microlides, and these would be like clarithromycin and then erythromycin. So looking specifically at drugs that can block these metabolic pathways, this would be figure 12.11, I'm sorry, the structures of some sulfonamides. And so the boxed side chains attach the ONOH group, right, of the nucleus, and then this one would be the sulfa actamide, this is the sulfadiazimide, and then this is the oxazole. Okay, so most of these are synthetic, which means they are man-made, and they are important sulfonamides or sulfa drugs. And this is the first antimicrobial drugs that have been made. They are a narrow spectrum, but they block the synthesis of folic acid by the bacteria. So you have your sulfazoles, and this would be like to treat UTIs, protozoan infections, etc. You have your sulfadiazones, and these would be like your burns and eye infections. Then you also have your trimethoprims, and this would be given in combination with the sulfas for UTIs and PCPs. We also have newly developed classes of antimicrobials, and these are formulated from pre-existing drug classes. And there are 3 different new types, and this would be the phosphomycin, trimethylthiamine, the synarchid, and then the daptomycin. And so the first one is a phosphoric acid, and it is effective as an alternative treatment for UTIs, and it can inhibit cell wall synthesis. We have the synarchids, and these are effective against the Staphylococcus and Enterococcus, and these that can cause endocarditis and other surgical infections. And when used, the bacteria are going to be resistant for other drugs, and because it also inhibits protein synthesis. And then the last class, the daptomycin, these are directed mainly against gram-positive bacteria, and they work by disrupting the membrane function. We also have ketolides like Ketex, and this is a new drug with a different ring structure from the standard erythromycin, and this is used for infection when it is resistant to other macrolides. We also have like Zyvox, and this would be a synthetic antimicrobial that is going to block the interaction of messenger RNA and the ribosome so that the protein can't be made. So these are used to treat methicillin-resistant Staphylococcus aureus, like MRSA, and then vancomycin-resistant Enterococcus like different agents that can be used to treat fungal infections. This is figure 1212. And so fungal cells are eukaryotic, and so a drug that is toxic to fungal cells are also toxic to human cells, so you have to use them carefully. And there are 5 antifungal drug groups. And the first one would be the microlide proline, and this would be like Tetracycline B, which would mimic or copy lipids. These are most versatile and effective. It's topical, and it is also a systematic treatment. And one example of that would be nystatin, and this is a topical treatment. So in this one here, this is the polines, and the example shown is going to be enterotoxin. We also have 5 antifungal drug groups, and these are shown here. The first of the 5 antifungal groups would be griselfulvin, and this would be used for stubborn cases of dermatophyte infections, and it is also nephrotoxic, which means it can attack your kidneys. You can also have synthetic azoles, and these are broad spectrum, like ketoconazole, I'm not a pharmacist, sorry, clotrimazole and microazole. And you also have the fluctosolides, and these are the analogs of the cytosines, the cutaneous mycoses, or they can be used in combination with amphotericin B for systemic mycoses. And then the last class would be the echinocannabinoids, and these can help damage the cell walls, and it is a capsofungin. And so these would be, again, different examples of this. This one here is the fluctosolides, and this is a structure analog of the cytosine that is going to be inhibiting DNA and protein synthesis. What about the antiparasitic chemotherapy? So we have antimalarial drugs such as quinone, chloroquinone, primaloquinone, and mefloquine, and we have antiprotozoal drugs such as metronidazole, the quinosides, sulfonamides, and tetracyclines. And then you can also have antihistaminic drugs that are going to immobilize or disintegrate or inhibit metabolism. Okay, and these would be some examples of these. Mebendazole, thiabendazole, and these would be the broad spectrum that are going to inhibit the function of the microtubules. They're going to interfere with glucose utilization and disable them. We also have the parental, and these can paralyze the muscles. They also have the nicotinamide, and this can destroy the scolex. Okay. When we're talking about antiviral chemotherapeutic agents, we have a selective toxicity that is almost impossible due to the obligate intracellular parasitic nature of viruses. And so what it's going to do is it's going to block penetration into the host cell. And by this, it's going to therefore block replication, transcription, or translation of the viral genetic material. We have those nucleotide analogs, which would be the acyclovir that can attack herpes. You have the ribavirin, and this is a guanine analog, and this can help treat RSV as well as hemorrhagic fever. And you have AZT, and this would be a thymine drug, and this is what's used primarily to treat HIV. If you ever watched the movie Dallas Buyers Club, they talk a lot about AZT when it first came out. We also can prevent the maturation of viral particles, and we do this by using protease inhibitors. And again, this is going to be specifically for HIV treatment. So what about drugs that specifically treat the flu or influenza? So this is going to be figure 12.6, and we have amantadine and rimantidine. And these are restricted almost exclusively to influenza A viral infections, and these are going to prevent the fusion of the virus with the cell membrane. You also have Relenza and Tamiflu, and these are slightly broader spectrums, and these work by blocking the neuraminidase in influenza A and B. And so basically these are the different drugs, and it tells you exactly where it's targeting the different receptors. And again, you can read more about that in figure 12.6. What about antiherpes drugs? Well, many antiviral agents mimic the structure of nucleotides, and therefore they compete for the sites on the replicating DNA. A couple of these would be like acyclovir and Valtrex and Famivir and Denavir, and these are both oral and topical treatments for oral and genital herpes, as well as chickenpox and shingles. And again, you can see how this is inhibiting that and therefore working. Again, that is figure 12.6. Moving on to drugs for treating HIV infections and AIDS, we have retrovirus, and these offer two targets for chemotherapy. The first one is interference with the viral DNA synthesis from the viral RNA, and this uses the nucleoside reversible transcriptase inhibitors, which would again be those similar shape of the nucleotides, so that's how they're going to interfere with it. And the interference then, because it's going to block this, is going to be interfering with the synthesis or the making of DNA using non-nucleoside reverse transcriptase inhibitors. AZT, like I said, is a very popular drug that's used widely now for HIV/AIDS treatment, and this was the first drug that was aimed at treating full-blown AIDS, and it is a thymine analog, so that is how it works. So this is going to be, again, that figure, which is 12.6. And this is going to be how the anti-HIV drugs are working and acting on different parts of the cell itself. Moving on to interferons. Well, these are human-based glycoproteins that are produced primarily by fibroblasts and leukocytes, and therapeutic benefits include, but are not limited to, reducing healing time, as well as reducing some complications of the infection. We can also prevent and reduce the symptoms of the colds and papillomavirus. It can also slow the progression of certain cancers like leukemias and lymphomas, and it can be used as a treatment for hepatitis C, genital warts, and Kaposi sarcoma. So how do we go about getting drug-resistant drugs? Well, basically, or bacteria, not drugs. This is an adaptive response in which the microorganisms begin to tolerate the amount of drug that would ordinarily be inhibitory. And this is due to either genetic versatility or variation, and it can be both intrinsic, which is like natural, or acquired, so it can be evolutionary. And a lot of this can happen because people don't take the rest or their entire, antiviral treatment, antimicrobial treatment, right? So you can build up that antibiotic resistance from that. So with this, we have the acquisition of it, and this is acquired resistance, and this is figure 12.13 in your book. So acquired resistance would be a spontaneous mutation that happens in critical chromosomal genes. And so we acquire these new genes or this sets of genes by transferring from other species. So this originates from resistance factors such as plasmids, or it can be encoded with drug resistance such as transposons. And so in this picture here, the bacteria transmit R factors, which often contain numerous genes for drug resistance by means of conjugation, transduction, and transformation. The widespread occurrence of such exchanges accounts for the rapid spread of the drug resistance. So here is another bigger table of the mechanisms of drug resistance, and this is table 12.14. And so up here you have the inactivation of a drug like penicillin by penicillinase, and this is an enzyme that cleaves or chops off a portion of the molecule and renders it ineffective. So that's shown there. And this one here, the receptor that transports the drug is altered so that the drug can no longer enter the cell. We have the third way. Okay, so this one, I'm sorry, was drug inactivation. 2 was decreased permeability. 3, here is the activation of the drug pump. And so what happens is that specialized membrane proteins are activated and continually pump the drug out of the cell. Over here in 4, we can change the active site. Okay, so we change in drug binding site. And so the binding site on the target is no longer the ribosome, for instance, and it's altered so the drug no longer can take effect. And then the last one is the use of an alternative metabolic pathway. So you can see instead of going straight, it jumps down here, right? And so this drug has blocked the usual metabolic pathway, which is green, that top one. And so the microbe circumvents it and gets around it by using an alternative unblocked pathway that achieves the required outcome, which would be the red workaround there. So what about natural selection? Okay, what happens naturally? So large populations of microbes likely include drug-resistant cells due to prior mutations or the transfers of plasmids. And there's no growth advantage until it is exposed to the drug. And then if it is exposed, then the sensitive cells are inhibited or destroyed while resistant cells will survive and therefore be left to replicate, otherwise known as proliferate. Proliferation is just cell growth. And so then eventually the population is going to be resistant, and this is from natural selection. Okay, so this is figure 12.15 in your book, and so basically over here you have generally any population of microbes will harbor a few cells carrying the prior mutation that confers the drug resistance, which would be these pink guys here. And then in B, environmental pressures. So for example, in this picture, exposure to the drug. This is going to select for the survival of these resistant cells. And then in C, in time, the resistant cell becomes dominant. Okay. So how do we limit this? Well, take your antibiotics, guys, and figure out do your whole course of treatment. And so this is in the newer version of the book, it is figure 12.8. And so we have usage, research your drug, make sure that you're using it for the right antibiotic, right, or the right cell that you're targeting. And then also we have long-term strategies such as reducing the abuse of antimicrobial usage. So what about interactions that occur between the drug and the host? Well, it's estimated that 5% of all persons taking antimicrobials will experience a serious adverse reaction to the drug, and that would be like the side effects. So major side effects can include direct damage to the tissue due to the toxicity of the drug, allergic reactions, as well as disruption in the balance of normal flora, and this would be like a super infection that can occur. Okay, so this is figure 12.16, where this is an example of a kid that was given tetracycline, and they have permanent teeth issues. They're going to be brownish teeth from enamel, just discoloration, decoloration, and that is going to be permanent. Then we have super infections, and this is figure 12.17, and this is the role of antimicrobials in disrupting microbiota and causing these super infections. And so a primary infection in the throat could be potentially treated with an oral antibiotic, and that would be over here. Well, then the drug is carried to the intestine, and it's going to be absorbed into the intestine, right? and therefore go into the circulation. And then the primary infection here is going to be cured, but now there are drug-resistant pathogens that have survived, and they can create an intestinal superinfection, so the next time you get at that antibiotic is not going to be effective. So what we have to consider when selecting an antimicrobial drug is we need to identify the microorganism that is causing the infection. We have to test the microorganism's susceptibility or sensitivity in various drugs in vivo or in vitro when it's indicated. And then also we need to understand the overall medical condition of the patient. How do we identify these agents? Well, identification of the infectious agent should be attempted as soon as possible, and then the specimen should be taken before the antimicrobials are initiated so we can see which antibiotics to use and which ones will be most effective. So how do we test for drug susceptibility? This is figure 12.18 in your book. And this is basically essential for groups of bacteria that are commonly shown resistance, and we can use this Kirby-Bauer disc diffusion test. And so what happens here is over in A, this is an example and evaluation of an agar diffusion sensitivity test. So if the test bacterium is sensitive to a drug, the zone of inhibition develops around the disc. The larger the size of the zone, the greater the bacterium sensitivity is to that drug. So the diameter of each zone is measured in millimeters, and then it is evaluated for susceptibility or resistance by means of a comparative standard. So you can see this is not very receptive to that antibody, but this one is. The circle is very, very large. So this one would be the most effective antimicrobial. And then in B here, you have the result of a gram-negative Kilbacera pneumoniae, when there are zones of inhibition that indicate the sensitivity to the CTX and ampicillin and the claravonic acid. And you can observe the expanded zone between these two areas here that indicates their enhanced action, and that would be that synergy that we were talking about before. And then down here, you can see that this is the results for gram-positive Staphylococcus aureus using gentamicin left and ATM on the right. Although the bacteria are sensitive to both drugs, the GM on the left, the gentamicin, has a few colonies growing in the zone of inhibition, which therefore, you can see these little dots here, indicates that there is some antibiotic resistance that has already developed in that concentration of the drug. Okay. So how do we do this testing for drug susceptibility? This is figure 12.19. And so this is called an E-test, and this is a quantitative method for determining sensitivity. So this is essential for groups of bacteria that are commonly showing resistance. And so we can either use that Kirby-Bauer that we just talked about in the previous slide or this E-test diffusion. And so with this, what happens is this test uses a plastic strip that contains a pre-measured gradient of the drug, and it's labeled with a scale that indicates varying concentrations in micrograms. The concentration ranges from higher at the top of the strip, up here where there's the zone of inhibition, to lower concentrations towards the bottom. When the strips are placed on a plate, swabbed with a test microbe and incubated, the overall area called the ellipse, right there, okay, of inhibition develops adjacent or next to the strip. Then the lowest point on the scale where the ellipse begins to form corresponds to the minimum inhibitory concentration. Okay, and so that would be there. And so, yeah, so this is the E-test, and so this is how we can use them to test even more sensitivity. Okay, so we can also do dilution tests, and this would be determining the minimum inhibitory concentration, or MIC, and this would be the smallest concentration of a drug that visibly inhibits the growth. Okay, so this over here is going to be figure 12.20, and this is a tube dilution test for determining the minimum inhibitory concentration. And so this antibiotic is diluted through tubes of liquid nutrient so that the concentration is successive in tubes, and it ranges from 6.4 micrograms per milliliter. Oh, this one's a little bit different, up to 1 milligram. Okay. And so all tubes are inoculated with the identical amount of test bacterium and then incubated. First tube on the left is the control that lacks the drug, and it shows cloudiness, which means that it is inactive, indicative of normal growth. And then reading from the left to right, the MIC is the dilution in the first tube in a series that has no growth, or therefore is not cloudy. Okay. In this one over here, this is a microbroth dilution in a multi-well plate that is adapted for pathogenic yeast. And this is figure 12.221. And so drugs that are being tested are Amphotericin B, which is AMB, then 5-FC, Fluc, and a couple other drugs, right? And so the pink indicates that there is growth, okay? Blue indicates that there is no growth. So reading across from numbers 2 through 12 over there, this is the increasing concentration of the drugs in micrograms per milliliter. 1 over here has no drug, okay? So that would be your control. So the first solution with no growth is going to be crossed out, indicating the MIC for that drug. So the results are given in the box on the left over here. Okay. And then the last slide. So using this, the in vitro activity of a drug is not always correlated with in vivo effects. So in vitro is outside of the body, in vivo is inside of the body. So if the therapy fails, then a different drug or a combination of drugs or a different administration has to be considered. And so it's best to choose a drug with the highest level of selectivity, but the lowest level of toxicity, and this would be measured by the therapeutic index. And that therapeutic index is the ratio of the dose of the drug that is toxic to humans as compared to its minimum effective dose. And so a higher index is going to be desirable. And that concludes today's lecture. We'll see you in class.